Hello, everyone. My name is Chinmoy Saha. I'm a senior technical support at Novogene Europe, and I will be the host of this webinar. I'm delighted to see you so many of you are able to join us today. Today, my colleague, Global Product Director Dr. Willie, and Product Marketing Manager Arman Mehra will go through a beginner guide to proteomics using Olink panels. But before that, let me briefly introduce our company. Novogene was founded in 2011 and has been growing rapidly since then. We are continuously developing our portfolio of next generation sequencing and third generation sequencing. And today we are present with services in various fields of science, from research, oncology, infectious diseases, and also food and water testing. Novogene is a global company with local contact person and sequencing facilities in Europe, Asia, and the US. We have over 2,600 employees and more than 5,600 customers. Our first European sequencing center is located in UK in the Cambridge Science Park. The UK lab is equipped with Illumina technology and our patented intelligent sequencing platform called Falcon 3. The automated workflow of Falcon enables us to decrease the turnaround time, minimize the factor of human error, thereby yielding consistently high quality outcome and leading us to process large amount of data. Additionally, we have launched another next generation sequencing lab in Munich, Germany. The Germany lab is becoming more operational with Novosec X Plus and PacBio Revio sequencing facilities. We provide a full range of next generation sequencing services from human and plant animal DNA sequence, various types of RNA sequencing, metagenomic services, and epigenomic services. Why Novogene? There are many reasons why you should choose Novogene as your sequencing partner. Providing consistently high quality service is our priority. To achieve this, we perform multiple quality control. There's sample QC, library QC, finally the data QC. We have large capacity, which we are increasing all the time. Recent example of this is opening our German lab facility using our patented Falcon 3 technology, which has not only a positive impact on quality and the capacity, but also turnaround time, since Falcon can run 24/7. We also offer customizable end-to-end solution. This means we can process your project from extraction to analyze the data. But you can also send us the extracted DNA and RNA or pre-made libraries and choose only receive raw data. Our product management team and labs are continuously optimizing protocol and services to ensure we have a sequencing solution that fits your needs. Lastly, our multiple sequencing facility equipped with Illumina, PacBio, and Oxford Nanopore technology give us largest sequencing capacity in the world. So we are very proud to support researchers worldwide in their work, and today we have a clear record in the scientific community. We have been cited over 16,000 research papers with a total impact factor more than 4,019. We are also driving the next generation sequencing industry forward with more than 60 NGS related patents. Your contact person are present in almost every European country. Get in touch with your local representative today to discuss NGS solution for your project. You can contact them directly or reaching out via our website. We also regularly host seminars in the university and various institutes across the Europe. If you would like us to organize a seminar near to you, please don't hesitate to get in touch with us. I would like to briefly introduce you our talented European sales team. They are based in different regions across the Europe, like England and Ireland, Germany, Southern Europe, Nordics, Eastern Europe, and Benelux. Our European sales team functions as your first contact person or contact to Novogene. So please don't hesitate to contact us with any next generation sequencing related request. Thank you for listening. That's all from my side for now. Now I will hand over the topics to our global product director, Willie. It's your turn.
Okay, thank you. Let me share my screen. Okay, can you see it?
Yes.
Okay. Yeah, okay. Good morning, everyone. Thanks for joining us for this webinar. And for today, we're going to have the beginner's guide to proteomics using Olink panels. Oops. So in this webinar, we'll have three parts. First of all, we'll have an overview of the proteomics techniques. And in the second part, we'll go over some of the Olink panels. And in the last part, we'll have some publications and references to show you how you can use Olink for your projects. So let's start with the first part, an overview of the proteomics techniques. I think the first question we would like to discuss is why we care proteomics so much. Because it is a direct measurement of biological activities, and also it has a very dynamic changes, which allow us to monitor our disease progression and also how we respond to different treatments. However, it will additionally, it will also carry a lot of most translational modifications. This information cannot be detected through some genetic biomarkers, such as when you do the WGS or the RNA-seq. And it's very easy for us to collect non-invasive body samples, such as blood and urine. So that will help us to simplify everything. And if you check the number of the FDA-approved drugs, most of them are targeting proteins. But the bad news is we fully understand all of our proteins in our body samples. So as we mentioned, actually blood will be the very easy or primary specimen that we can collect to study our proteomics because it represents the largest and deepest version of the human proteome presence. So when you collect your sample, you can add in the anticoagulant and then you do the centrifuge, then you will either get plasma or serum on the top. So with that, it will be very easy for us to process your samples and perform the experiment for protein quantification. So basically, according to the publications from Olink or by using Olink technologies, most of them, I mean, the more than 80% of our samples will be plasma or some other sections of the blood. But even though we are very easy to get our samples, but we have to we have to face this kind of challenge, which means it has a very dynamic range in the protein concentrations. So on the left, we're showing the plasma protein concentrations, and you will see a lot of the green part, which is the classical plasma proteins. So those have very high abundance, but if you move down, you will see the tissue linkage proteins and some second cytokines. So those will be labeled in yellow dots. So those will be very valuable biomarkers that we really want to monitor or check. So for those biomarkers, if you are using the traditional methods such as mass spec, it's very hard for you to quantify them very easily. So now with this kind of fact, like the majority of our proteins are in extremely low abundance. And if you want to study high abundant protein and low abundant protein at the same time or in a single run, it's very urgent for us to find a good technique to help us to fix this challenge or to to fix this kind of challenge. So now let's see if the technique is ready for us to move forward. Then we need to review the history of our proteomics. Basically, most of our studies are driven by mass spec or the immunoassays. If you can check the past decades of those kind of new technologies or instruments, so basically most of them will be mass spec and immunoassays. But once we moved to here, we do see some new players such as Olink and Somalogic. So they are proposing some new technologies to help us sequence or quantify the protein levels, thousands of protein levels in a single run. Here is an overview of this kind of proteomics techniques. So on the left, we show the mass spec. So this is still a golden standard for us to perform the untargeted proteomics. It allows us to discover new biomarkers or new proteins, but you have to have a very high expertise in this kind of area, and you also to perform a lot of sample processing or sample treatment before you load your samples on some instrument. And the cost for the instrument is also high. So if you would like to sequence hundreds of thousands of samples, it may take weeks to months. But the other one, the immunoassays, is another traditional method we will use for proteomics. For this one, it is targeted proteomics. However, we can only quantify or we can only study like 1 to 50 proteins at the same time. So even though it allows us to move a little bit further, but not that far. So that's why we now have this kind of high plexing technology. So this one allows us still a targeted proteomics, but in a single run, you can quantify up to 5K, 7K, or even 11K proteins in a single run in one of your samples. So with that, it speeds up everything. So if you want to study thousands of samples or hundreds of samples, you may finish your project within 2 days or even 1 week. So that allows us to speed up everything for the clinical study. And also it delivers very nice limit of detection. So we can detect as low as phytogram per milliliter for some of your biomarkers or proteins. So with that, we do see the advantage of high plexing techniques, which can allow us to move forward from research to clinical application. And it also helps us to shorten our study from decades to days. But actually, we will see that it is not a kind of the replace the traditional mass spec technology. It only complements our traditional ones as it is the targeted protein omics. And it will help you to study your well-known proteins very easily. So here will be some details about the mass spec and also the Olink. So on the left, we will see the general workflow of the mass spec. So once you have your samples ready, we can extract your protein, and then we do the depletion to remove the high abundant proteins, or we will fractionate our protein samples, and then we will quantify or we will measure the peptides by using the mass to charge value and to either identify a protein or quantify a protein. And then we will move forward with data analysis. So this is the traditional we will use for the mass spec. And on the right, we will show the details or the workflow of our Olink techniques. So still, we start with the plasma, but the good thing is you don't have to process your plasma or process your samples. We only load our samples onto the plate, and then we will add in the antibodies in the kit, and then we can do the overnight incubation that will allow us that will allow the protein to be targeted as shown on here. So here the blue dot will be the protein we want to target, and then in each of the kit you will find thousands of pairs of the antibodies, which showing here and this one. So those once a pair of antibody target this protein, and each of the DNA single strand DNA will bond with each other, will pair up with each other, and to form this DNA double strand DNA. And for us from here, we don't need to perform different biological process to quantify the protein. We simply translate our protein signal into DNA signal. So you can quantify this DNA amplicons by using either qPCR or using NGS technologies. So that will simplify everything for us to quantify our proteins. So here will be the general steps, as I mentioned. So you just collect your samples like a drop of your blood, and then we do the incubation, and then we do the amplification, and then we analyze the data with either NGS or qPCR. So that will be how the Olink work in all of our samples. So here will be a very brief overview of the Olink of the proteomic technologies, and then we will see what kind of panels Olink will offer. Basically, in the in clinical researches, as we mentioned, Olink is kind of proper for clinical studies. So we have different stages, which is showing on the top. So basically, at first, we will discover without a lot of hypothesis, but then once we find our candidates or the biomarker of interest, we will move forward for the preclinical research and then move forward to different phases of the clinical development. So on the bottom, we show different panels Olink can offer. So on the left will be the very large panels, including Olink Explore HT, which can quantify more than 5,000 proteins in a single run, and then Olink Review, which can quantify more than 1,000 biomarkers in a single run, with very tiny amount of input of your samples. And those will be performed on NGS platforms. And on the right will be some other smaller panels, such as the Explore 384, Target 96, Target 48, and those two will be some customized Olink panels. So for Novogene, we now currently offering Explore HT and Review, as we think now we are still in the discovery range or era of the proteomics. But if you would like to reach this kind of number showing in the middle for Explore 384 or Target 96, you have to perform the experiment on multiple panels as shown here. So basically, once you perform 4 panels, that will give you more than 1,000 proteins. And if you perform your experiment on 8 panels, that will allows you to move forward to 3K of your proteins. So this is a very brief overview of other smaller panels. And back to here. So basically, we majorly use Explore HT and Review to support our discovery stage, either broad discovery or the hypothesis-driven discovery. And later, once you move to validation and development in your clinical study, then the smaller panels, such as the 96 or 48 or some other customized panels, will be more suitable. So those panels will be more like disease or process focused. Then what's the difference between Olink Explore HT and Review? Because those two can be used for discovery for different objectives. So first of all, that will be the numbers of proteins we can detect. With HT, that will be more than 5,400 proteins. While for Review, it's more than 1,000 more 1,000 proteins. And the sample volume for each reaction is also different. HT, we need a smaller sample volume, like 2 microliter, and that will be 4 microliter for Review. So for the proteome coverage, it's also different. At the top level of the reactome pathways, both of them can give you 100% of coverage. However, if you move down to some move down to other levels of the pathways, HT will give you very high coverage, like 85 of all pathways in reactome, but for Review, that will be only 64. And for Review, it is actually developed by studying all of the Olink previous projects, and they pick up some very good biomarkers or very robust biomarkers. So those biomarkers are usually focused on or fostering the inflammation or immune responses. So if you check if you want to perform the deep profiling of inflammation or immune response, then Review probably will be a better choice because it can give you 96% of the coverage. However, if you are not satisfied with this kind of coverage, you can definitely move upon to the HT panel. So this will give you a very nice coverage up to 99% of those kind of immune response pathways in reactome. So with those kind of different parameters, we do have different applications by using those two panels. So for HT, it is very well, it is very nice suitable for the cohort study with this highest chance for you to discover very new biomarkers or cis-pQTLs. So that could be possible diagnostic biomarkers or some new drug targets. And for Review, so even though you can only quantify 1,000 proteins, but still it is a very nice budget friendly screening tool for your for you to discover new biomarkers. And if you really want to do deep profiling for the inflammation, so Review will be the first choice as well. So a little bit more details about HT and the Review. So here is one page for Explore HT panel. If you compare the human proteome from the Human Protein Atlas versus the Explore HT, we will see a very similar distribution across the intracellular proteins, secreted proteins, membrane proteins, and some other isoforms. So that's why we Olink developed this kind of HT that will help you to do a very broad targeted proteomic studies for you to discover some biomarkers or some drug targets. And on the right, it listed the number of different types of proteins, including the inflammation, secreted, tissue-specific drug targets, and also some exploratory proteins. So this HT panel, if you are following the UK Biobank news, you will find their announcement in the early this year. I think it's in January this year. So they already started their phase 2 cohort proteomic study. So they are going to use HT panel to run this kind of panel on 600,000 samples. So this is a huge project, the world's most comprehensive study of the proteomics in a very large size of the samples. So to make this decision, before they make this decision, they also started with a pilot study in 2023. So in that study, as HT is developed in 2023, so before that, they are using the 3K panel to run this pilot study. And from this pilot study, they performed the proteomics on, I think, will be 54,000 samples. So with that, they identified pathways from drug targets, they improved the disease prediction scores, and they got more than 100 peer-reviewed studies publications. And they identified more than 14,000 of PQTLs. So most of them, more than 80% of them, were previously unknown. So those kind of new identified biomarkers will support a lot of disease studies, such as the heart disease, the Alzheimer's disease, and other neuro disease as well. So those will be very good data for all of those regions to use and to share. So that is the some details about, that will be some details about the HT panel. And on this page, those will be the details for the review panel. So for this one, as we mentioned, it includes more than 1,000 proteins for broad coverage of the proteomics. And it is very good design, well designed for the deep profiling of inflammation process for disease research. So for this one, you will see still the number for different categories of proteins, such as inflammation proteins, the proteins associated with disease, the proteins associated with cis-pQTLs. Because for cis-pQTLs, it's quite important for us to associate genetic information and also the proteomic information. And those will be very important for further disease studies. So as we mentioned in the details for the coverage, for the top level, both of them can cover 100%. And for all levels of pathways, so review is a little bit lower, like 64%. But for inflammation, if you want to do deep profiling, so it can also deliver very nice coverage. So here will be some details for the coverage at different levels of our pathways. So now I'm using the reactome pathways coverage figure to show you how we can visualize the difference. So this is the pathways Explore HT has covered. So if you see all of those yellow lines, those will be some target proteins those will be some pathways the protein in the HT panel have covered. And if you see some white lines, those will be the pathways we cannot cover by the Explore HT panel. But if we add the review on the top layer, so those kind of pink or purple-ish lines will show how review cover those pathways. And we do see a little bit difference in some of those kind of major biological processes. So you'll find more details if you log in the Olink Insight. You can see more details and zoom in this kind of cluster to see which pathway has not been covered by review and which pathway is only covered by HT. In addition to those kind of pathway coverage, so we also see a difference in the sample volume per run. So for HT, we need a little bit lower, but for review, we need 4 microliter. And for the sample types, which is compatible to Olink service or Olink panels, here will be the list. And on the top, that will be the standard metrics or the sample types we can accept, including human plasma and human serum. And for the bottom section shows the list of alternative metrics. So for those, you may follow some instructions showing in the Olink website, like telling you how you can process your samples, and then you can send your processed samples to us. Then we can move forward with this kind of incubation and sequencing and then data analysis. So if you have no idea what kind of how your sample will work with the Olink panels or service, you can go to Olink website, and then in this kind of knowledge section, you will find a publication in the drop-down list. So with this kind of window pop up, you can type in the keywords of your study, such as sample types, such as disease name, such as the protein name, or you can select your sample type in this kind of drop-down list to see if there is any reference for us to follow to start your new projects. So that will be the kind of overview of some of our panels. And then we move to the last part of the application. So actually for Olink service, as we mentioned, it's very suitable for the clinical studies. And for those kind of studies, we can group them into 3 different applications. So first of all will be the protein biomarker from discovery to implementation. And those will fall in scenarios such as disease screening, diagnostics, risk stratification, or disease mechanism research. And the second group will be the drug MOA, the mechanism of action. So that will help us to identify novel biomarkers and drug MOA insights from clinical trial samples. And the last application will be the biobank or cohort proteomics. So it is very similar to the UK biobank study, like you perform the population proteomics, which can empower your population genomics. So those will be some other scenarios you can follow or you may occur when you design your projects. About which panel is the perfect one or the best one for your study, it really depends on which stage you are in when you move forward. Because as we mentioned for HT and review, it is quite well designed for the discovery, either for broad discovery or for the hypothesis-driven discovery. And for other smaller panels, those will be more suitable for the validation or the development stage. So let's see more cases or references for each group of the applications. This is the first one, that is the biomarkers from discovery to implementation. So basically we will follow the roadmap on the left. So we start with the biomarker screening, and then we validate the biomarkers or the candidates we identified from our study. And then we will perform or we will develop a diagnostic model by using bioinformatic analysis or bioinformatic tools. And with that, we will follow up our cohort or population to collect more information to see if our model is suitable or not. And with this kind of more information collected or more sample has been collected, we can perform the prognostic analysis or risk prediction by using all of those informations. So basically, when you set up your study, we will recommend 2 different methods or common methods for the biomarker screening and validation. So in the middle, we show the triangle method. So you can start with very small cohort, but you have to combine with a very large panel, like the review panel or the HT panel. And with that, it will allow you to have a very nice discovery or screening of the potential biomarkers. And then you can move to the verification stage by using a moderate cohort size, and then combine with a smaller panels, such as the Target or Explorer 384. And then that will work well for the verification. And the last step, that will be a validation step. You can use very large cohort and very large population, and then combined with either customized panel or immunoassays for the validation. Because from here, starting from the very large panel to those kind of very few candidates, now we don't need to run with very large panel again. So that will help you to narrow down your candidates very nicely by using this kind of triangle strategy, starting with the smaller cohort, large panel, then move down to the large cohort together with the immunoassays or customized panels. So this is the triangular method. And the other one will be the rectangular method. So in this one, we only need to set up 2 cohorts. So for first one is for discovery. So you start with a very large cohort, and then you use a very large panel for discovery to find which biomarker is very validated. So like in this one, or the blue one, or the red one. And then we run another cohort study. So this cohort should be very similar to your first cohort in the size-wise or in the kind of the parameter-wise. So that will be used for validation. And for here, we still use a panel with your candidates or with your potential biomarkers, and then for the validation. So by paralleling analyze your data from both cohorts, that will give you an overlap or validated biomarker, which only shows in red. So some of the green may specific to your discovery cohort, and some of the green may only belongs to the validation cohort. But the very nice biomarker you want to move forward will be the red ones, which has been validated in both cohorts. So that will be another way for you to set up your cohort study or for the biomarker study. And in the following several pages, we will show you some cases. So this is a reference from the Alzheimer's disease study. For this one, they used the triangle method. So they started with a discovery cohort, like 180 samples, and they are using combined with more than 1K plasma proteins to perform the discovery step. And then they found out there will be 492 differentially expressed proteins. And then they perform some the bioinformatics analysis. So that is a customized bioinformatic analysis to narrow it down from more than 400 to 19 hub proteins. So they found 19 clusters, and in each cluster, they pick up 1 hub protein to perform the validation. So in the second round of discovery, they only use the bioinformatic results by using the 19 hub proteins to perform the discovery cohort again in the 180 samples. And that is the second round of the discovery. And then they move down to the final validation cohort by having almost 100 samples and still using 19 hub proteins to perform the validation. And with that, they found those 19 hub proteins performed very well in the AD classification. So this is a figure it shows in the publication. So this kind of AUC curve will show how well your biomarker will work for this kind of disease study. So the higher, so if the higher, the better. So sensitivity is good and specificity is good. So the area below this kind of curve shows how well your biomarkers will be. So this is for the AD study. And the next one is for the multiple sclerosis study with Olink. So this one, they are using the rectangular method. As we mentioned, so you are going to use 2 similar size of your cohort. One will be the 150 cohort by using 1,400 proteins. And in the validation cohort, they are using 71 samples, still 1,400 proteins. And in this one, they discovered 42 differentially expressed proteins. And this one, they released 25 differentially expressed proteins. And those 2 cohort studies can be validated with each other by analyzing parallelly. So on the top, it shows how they perform the whole process. So they set up their cohort here with different different genders or different sample types. In this one, they, in addition to use plasma, they also use CSF for another type of samples. And then they use the Explore panels to move forward. And from this kind of study, you will see the major results on the left. So the NFL will be a very nice biomarker for them to detect after they collect the samples for 2 years. So that will be a very nice biomarker for you to perform the disease study. And then they also pick up 11 biomarkers from here to here. And also they normalize the age data into this kind of model to make this kind of N-arms model, N-arms score to use this N-arms score to help you to distinguish the disability worsen level and also to perform the diagnosis of the MS disease. So this is another paper we showed to use the different Olink panels to perform the biomarker study. Because Olink HT and Olink review panels were developed or launched very recently. As I mentioned, HT was launched in 2023, and the review was launched in 2025, this year. So that's why we see very few papers or publication on those two panels. But we do have some cases. Here is one for HT. So they started with 2 still discovery cohort and validation cohort. And by comparing HT versus 3K panels, the previous version of the large panel, they do see very unique biomarkers in HT. And then they do see very nicely increased statistical significance when they use the HT by comparing to the previous version, which is the 3K panel. And this is a case for review. This is a case study for review. So in this one, they started with 500 individuals, and then they identified 22 acids, which were significantly associated with the glucose tolerance. And then they proposed a new hypothesis, and now they are following this hypothesis and to move forward. And by collecting samples at different time points, they found out the overlap between different time points for the biomarkers and also some unique biomarkers at different time points. So here will be some major results in these cases. Because review is very well picked based on previous studies, like some well-performed acids or proteins or very robust proteins. So we do see relatively higher level of detectability and very nice precision in this kind of review kit. So for the second type of application, that will be the novel biomarker and drug MOA, which is the mechanism of action. So in this kind of application, we usually set up a longitudinal cohort, which you can collect your samples at different time points after you make the treatment or you use the drug. And then, or we can use some samples from previous clinical trials. So those will always fall in tumor samples, rare diseases studies, or metabolic studies. And with those kind of study, they usually combine Olink data with other omics data, such as WGS, RNA-seq, or single-cell-seq, or even some ELISA results to validate their biomarkers or their mechanisms. And then they move down to the they probably will get some new biomarkers, and then they will clarify the mechanism of the action, and then they probably will develop a predictive model as well along with this kind of circle. So still some cases for different from different publications. So this one is a phase 1 clinical trial for melanoma. So this one, they use 14 patients, and they perform the first line therapy and to see how those therapy will affect the immune system in those kind of patients. So in this kind of study, they combine the Olink together with single-cell RNA-seq and also T-cell receptor sequencing. So those results all deliver those kind of techniques or deliver the transcriptome or proteomic changes during the therapy. And they also find some very good biomarkers for them to clarify the mechanism of the action after this kind of treatment. So in this kind of second reference will be a phase 2 clinical trial by combining the Olink and set-off techniques. So this is for a bladder cancer, still a study on different, like around 70 patients. So they collected different tissues, including blood and urine, and they also perform Olink and set-off on different sample types. So in this one, as I mentioned, Review and HT are only recently launched, and previously they can only use the smaller panels to clarify the MOA or for the drug responses. So at that point, they are using one of the target 96, and those kind of results demonstrate a collaboration between our immune and clinical outcomes. So here, they collect the samples at different time points and identify some different biomarkers by comparing before or after the treatment. And also this kind of biomarker help us to differentiate how long they can survive after the treatment. So again, so for HT and the review panel, we got very few cases. So this is one for the HT publication. So it's a CAR-T therapy in the AML study. So in this one, again, they found very similar results. For some of the factors, we do see increase in their levels. And it will also allow us to find new factors such as the IDO1 and IL-3 in this kind of study. And this is another one by using review kit. It is also a case study delivered by Olink and their collaborators. So with different drugs or different treatments, you will see the biomarker has been changed very differently for different patients or at different time points. So the last one will be the population proteomics. So this one, it is very commonly see the Mendelian randomization and the pQTL study is very commonly seen in this kind of publications. So on the left, it shows how the pQTL can support your further study or further application of your academy research results. So on the right, it kind of shows you the possible results by combining genetic information versus the protein information. So the genetic information will be our GWAS or genetic variants that we collected from the WGS or GWAS study. And then we associate or collaborate it with the pQTL, which is the protein level QTL biomarkers. And if you check on the right, there will be some possible results. If we find one of the variants will lead to some change of the protein and then further lead to disease, we will call this as the causality result. And those proteins will be our potential drug targets, because once we target it, we probably will block the disease or we will avoid the disease from the very early stage. And the second will be the reverse causality. So this one, once we see multiple or one of the variants, you will find the disease occurred first. And this disease will lead to some change in the protein level. So in this kind of situation, our protein will be the potential biomarkers that we can use to monitor our process of the disease. And for the rest, the third one is the horizontal, the third one. In this one, with one of the variants, we will see protein will show up and disease will occur, but we hardly find the connection between proteins and disease. So this one, we don't have, actually, it doesn't deliver a lot of useful information. And the last one as well. So this one, with the multiple variants. One of the variant will lead to protein change, and the other variant will lead to disease. And still no connection between protein and disease. However, we may see some linkage between those two variants. So this probably will result in some false positives for disease and proteins and disease. So basically, it's better if we can find the result one, the type 1 result or the type 2 results. That will allow us to move forward with some clinical trials. And here will be two examples for this one. Is kind of discover novel drug targets by combining the two cohort study. One is from the CKB and one is from the UKB. So by combining the data from two cohort data set, and also by combining Olink and GWAS study results, so they will be able to identify 4 proteins as the protein targets. So that will be the potential drug targets for us. So that falls or agrees to the result, type 1 result. And the other one will be the risk factor screening. So this one, they still use the different cohorts in discovery and in the validation stage by using different sizes. So this one, they set up the 80 samples for the discovery cohort, and they use the UKB biobank data as their validation study, still using Olink Explore panel and combine the Olink and GWAS study results. With that, they find the evidence as well to find out some biomarkers for them to perform the risk prediction and also for further drug development. So with that, I think I'm done with that kind of applications to show you how HT review or other panels to support different types of projects. And I will pass to Armin.
Hello, Wei. Thank you for your presentation. I'd share my screen now. I'm having some troubles sharing my screen for some reason. All right, I hope you can see the screen now, and I hope it's in the right view. Miranda, can you give me a confirmation for the same?
Yes, but it's not in the presentation mode.
It's not in the presentation mode. Let me check. I don't know what's going wrong today. Do you see the right mode now?
No, really. We can see the PowerPoint, the left side slides. Yeah, right now it looks perfect.
Right now it looks perfect?
Yep.
Okay. Okay. So just confirming again, everything's fine, everything good for now. If I'm changing the slides.
Are you changing it?
Yes.
I can only see the
Okay.
front.
Okay, something is off. Maybe what I'll do is I'll remove my second screen and hopefully that should solve the problem. Extremely sorry for the
No problem.
Let's try again. I hope now it's okay.
Yeah, looks perfect.
Perfect. I'm extremely sorry for the confusion regarding the screen. So I'm Arman Mehra. I'm a product marketing manager at Novogene Europe. And I'm just going to expand on what Wade talked about. And I'm going to focus more on Novogene's service for Olink Explore panels, which be it Olink Explore HT or Olink Reveal. And I'll give you some more details on our service specifications. So currently, Novogene is offering two different kinds of panels from Olink. One is Olink Explore HT, which, as we explained, is for more than 5,400 proteins. And the other one is a new panel, which is Olink Reveal, and that covers around 1,000 proteins. Now, in terms of the final data output, both of them have similarities because the readout is going to be in the NPX format. And as we also explained, that these panels have a great dynamic range. So you can even detect proteins in the femtogram range. And also, it's important to mention that the ultimate readout for both of these panels is going to be NGS. So finally, we are going to read those barcodes or those DNA sequences using NGS. So talking a bit more about Olink Explore HT. So this panel measures over 5,400 proteins with proven specificity to gain understanding of your biological question at the protein level. So the advantage is that it has a broad dynamic range. So it allows the detection of low abundant proteins with ease. And also this panel provides high specificity, high coverage, as well as high throughput, and offers you reliable results. Finally, this panel offers comprehensive protein profiling by measuring thousands of proteins, which really helps us to uncover valuable biological insights. The second panel that we offer is Olink Reveal. This panel measures more than 1,000 proteins and offers a simplified workflow. And also, this has brought down the costs. Olink Reveal, it also offers a broad proteome coverage. So we get coverage of 100% top-level pathways and 64% of all pathways in the Reactome database. It is particularly helpful in profiling of inflammatory processes. So 537 inflammation proteins covering 96% of immune response pathways in the reactome can be studied with the help of Olink Reveal. Also, it covers more than 1,000 proteins, and some of these proteins form robustly detectable markers. So this is also really helpful for these proteogenomics studies. Overall, the workflow for Olink Reveal is simplified. It's a more cost-effective solution. And again, like Explore HD, it offers high specificity, precision, and reproducibility. Now, for Olink, Novogene offers end-to-end service for both of these panels. So we start with the pre-study discussion. You can reach out to us. We can talk about the projects, the sample requirements, and so on. Then we can actually help you ship the samples. Finally, we do the library preparation at Novogene, followed by sequencing, and we can also carry out the data analysis and deliver you the final results. I also want to highlight here that library prep does not just include the NGS library preparation, but also all the steps that are required to be performed for these Olink panels would be performed by Novogene. Now, giving you an idea of our sample requirements. So the primary sample type for Olink panels is human serum or plasma. These are going to be the two major sample types. And the sample amount that we recommend is that you ship us 50 microliters or more in 96 well plates. And these if you're sending the samples in 96 well plates, the plate should be well skirted. So the borders should be solid so that the plate can stay flat and so on. And this is also more protected. If you can't send them in plates, you can also use Eppendorf tubes. It's important to remember that the transport has to be with dry ice and frozen delivery to avoid sample thawing during shipping. Also, some instructions for storage would be that we recommend that you store your samples in minus 80 degrees Celsius immediately after sample collection. Also, it's important to remember that hemolysis may affect analysis results, so you should try to minimize that. Apart from human serum and plasma, it's also possible to accept other samples from human body fluids. It could be saliva, it could be some kind of cell culture lysate or cerebrospinal fluid. However, if you have these non-standard matrix types, then please reach out to us and we might have like some tailored requirements for these other sample types. Now, some particulars on sequencing. So for sequencing the libraries, we use NovaSeq 6000. The sequencing strategy is single-end 24, and we use a 35-cycle kit. The data amount that we sequence is going to be approximately 40 million reads per sample. Now, in terms of data delivery, you can either opt for without bioinformatics analysis. In this case, we just deliver you data in the NPX format, or we could provide you the full bioinformatics analysis with data in NPX format, differential expression analysis, enrichment analysis, and so on. So this essentially gives you an idea of our bioinformatics pipeline. So we have the data coming from the sequencer. These files give us idea of the counts, and this data is fed into the pipeline where it's converted into NPX data. So NPX stands for normalized protein expression, and it's Olink's unit for relative quantification of proteins. So we feed this data into the pipeline, we normalize it, we perform quality controls. Finally, we do quantification. If you have different sample types or conditions or groups, we can do differential expression analysis. And finally, we can do functional analysis to see which pathways might be affected. The data that we give to you is in the form of a report, and our report contains several different kinds of figures that help you visualize the data. If you want to have a look at the report, then please reach out to us, and our account managers will share a copy of a demo report with you. Finally, I would just like to mention that the analysis that we carry out is based on the guidelines from Olink. We use the pipelines recommended by the company to ensure that you get high quality protein data. So with this, I end my presentation, and we would be happy to take up questions.
Thank you, Arman and we, for nice overview of today's topic. So in the meantime, we received quite some question from the audience. So I would like to highlight some of the questions and provide the answer. But if we have time limitations, so if we are not able to answer it, we will answer it via email and contact later. So I can see the first question is, how does the panel work for discovery of new proteins if it is using already known antibodies?
Okay, I will take this one. So for actually each of those panel have a fixed list of proteins in this kind of panel. So we'll have this kind of well-designed antibodies to target this kind of fixed list of proteins. And this panel is not using to discover new proteins, it's using to discover new biomarkers in your studies, including some disease studies or drug responses studies. So it's not to discover new proteins. For new proteins discovered, is still we go back to mass spec.
Okay, thank you. The next question is, how do I know if my protein of interest is covered in the Olink Reveal panel?
I can take this up. Yeah,
Okay.
or where do you want to take it? Yeah. So basically, there's a list of proteins for Olink Reveal and Olink Explore HT. You can find the list on the Olink website, or if you cannot locate it, just reach out to us. We can share the Excel file with you, and you can find if your protein of interest is represented in the panel or not.
Thank you. The next question is, can you perform some custom bioinformatics analysis for me?
It really depends on what kind of analysis. But if you can talk to us and discuss what kind of process, like figures or pipelines you would like to include in this kind of analysis, we will have our BI expert to discuss with you and to see how we can deliver those data.
Thank you. And the next question is, do I need to send the internal and external Olink control myself?
Actually, no. So in each of those kind of panel keys, it will include internal control in those reagents and also external control samples in this kind of kit. So I didn't show the slides, but I can include that after the webinar when we send out the presentation slides. So they have very nicely designed internal control and external control samples in this kind of experiment.
Yeah, just to again highlight the point, those controls are from Olink. So those are going to be provided by Novogene. And of course, you just have to include the controls that are required for your study.
Yeah.
Thank you. The next question, how many PTMs are converted with the Olink panels? And can I detect PTMs or isoform using Olink?
Actually, for PTMs, definitely no, because that can be only detected by mass spec. And Olink is a technique for quantification of each protein. So it will be a very quick and easy way for you to quantify your protein of interest or targeted proteins. Yeah.
So basically, I have I've seen a lot of attendees. They are asking about can the panel is available for other species than human and mouse. Do you have any comment on this?
So actually, Olink is running this kind of internal test as well. But from one of our projects, HT can each of those kind of panels can be used for other species. However, the detectability will be pretty different compared to human samples. For humans, so it's possible for you to detect like 3,000 or 4,000 proteins, but for mouse samples, when you use UHT, it's usually like 40% of those proteins can be detected. So it really depends on how similar this kind of protein shared by mouse and human. So yeah, it's quite possible and worth to try with different, mouse species, but we cannot expect a very high detection rate. Yeah.